the previous video, we have seen the basic three models of a replication, that is semi-conservative, conservative and dispersive model. So now to prove this, which is the right model of replication, Messelson and Stahl started this famous experiment on DNA replication using the E. coli bacteria as the model system. The last video I have told you they specifically went on choosing nitrogen isotopes for adding in their nutrient broth, which they have used to grow E. coli. And I also mentioned why they specifically went for nitrogen, because nitrogen bases are present in the DNA. So now we will continue with this experiment. After many generations growing in the 15N medium, that is the heavy isotope of nitrogen, the nitrogenous bases of the bacteria's DNA were all labeled with this heavy N15 isotope. Then the bacteria were switched to medium containing the light, that is the normal N14 isotope, and it was allowed to grow for several generations. So initially they took the N15 medium and they grown the E. coli organism in the particular medium for many generations. So now the entire DNA of this E. coli is going to have this N15 medium. So after some time, once this entire DNA is going to have this N15, now they are immediately switching it to the next medium containing the N14 isotope. So in these two mediums, the only one difference is the addition of the nitrogen isotopes. In one it's heavy nitrogen, another one it's the light nitrogen isotope. Now DNA made after the switch would have to be made up of N14, as this would have been the only nitrogen available for the DNA synthesis. Now Messelson and Stahl, they knew how often E. coli cells divided. That's the one reason why they specifically went for E. coli cultures, because E. coli was the most studied organism at that point of time. So they were able to collect small samples in each generation and extract the purified form of DNA. They then measured the density of the DNA. So how they did this measure is like an indirect manner to check the content of N15 and N14 using the process called as density gradient centrifugation. This method they specifically opted because this separates the molecules such as DNA into bands by spinning them at high speeds in the presence of another molecules like cesium chloride. In the presence of the cesium chloride, it forms a density gradient from top to the bottom of the spinning tube. The density gradient centrifugations allows very small differences like those between N15 and N14 labeled DNA to be detected. This is the main reason why they opted for this method, density gradient centrifugation. Now finally they got all the samples collected at different generation intervals, and they observed the bands that appeared from these three things after performing the density gradient centrifugation. So the first generation, that is generation 0, where it was completely grown only in the N15 heavy isotope. The DNA isolated from cells at the starting of the experiment, which we call it as generation 0, because it's just before the switch to N14 medium. So that's the first step where we are starting our experiment. And the cells isolated from this produced a single band after centrifugation. This result made sense because the DNA should have contained only heavy N15 at that time, because it was not switched yet to the N14 medium. The second generation, that is generation 1, this is the first generation after switching to the N14 medium. So the isolated after one generation, that is one round of DNA replication, it produced a single band when centrifuged. However, this band was higher, intermediate in density between the N15 heavy DNA and the N14 light DNA. So now they got curious how this was like happening, but actually this was corresponding to one of the models of the replication. I will say what is that particular model. We will see what happened in the next generation. When second generation DNA was centrifuged, it produced two bands. One was in the same position as the intermediate band from the first generation, while the second was higher, appeared to be labeled only with N14 nitrogen, that is the lighter version of the nitrogen. So this is the diagrammatic representation of the Messelson and Stahl experiment. To observe this diagram very clearly, you could see this is the N15 medium, which is the heavy isotope containing medium, and this is N14 medium, which is the light isotope of nitrogen containing medium. So when the E. coli culture was grown initially in the N15 medium, the DNA of the E. coli is going to be of higher density because it was containing the N15 in its DNA. But when it switched to the N14 medium, now the newly synthesized DNA after every replication steps are going to have few traces of N14, though it's going to have N15, which is its parent DNA strand. So to identify it, they grown it for 14 generations. That is, after inoculating into N14, they checked out for 14 different generations of growth. And as I already mentioned, they know the time of replication of this E. coli culture, which was around 20 minutes in duration. So for every 20 minutes, the E. coli is getting replicated. So this is the result they have got after collecting the readings. So this is the 0th reading, that is the 0th generation, where the medium was completely N15 and it was not switched to N14 medium. So here you could see the density was like N15, so it was settled more like in the bottom level. But when in the it was switched to the N14 medium, now the newly synthesized strands are going to have N14. That's why you could see the single band that was formed was in between the lighter version as well as the higher version. When in the second generation, that is the generation 2, you could see 50% had the strands from this intermediate and another 50% was the strand from the lighter N14 medium. Then gradually in the generation 3, you could see the 75% was from N14 and 25% was from intermediate. So this level of progression clearly stated that the newly formed strand after every replication step is going to have the N14 medium and the another template strand is going to have from the parent one. So this proved that the Messelson and Stahl that DNA was being replicated in a semi-conservative manner. The pattern of two distinct bands, one at the position of a hybrid molecule and one at the position of a light molecule, is just what we'd expect for the semi-conservative replication. So this Messelson and Stahl experiment proved replication occurs by the process of semi-conservative replication.